This is the first Spotlight interview of 2023, and we have the pleasure to have with us Fábio da Costa, who has a bachelor's degree in microbiology and immunology, a master's degree, and a PhD in biological sciences, microbiology, all from the Federal University of Rio de Janeiro. He has experience in cytotoxicity and antiviral activities studies and molecular characterization of neglected species of rotavirus. In 2021, he joined as a postdoc in the project of genomic surveillance of SARS-CoV-2 in Rio de Janeiro state, conducted in collaboration with the State Central Laboratory of Public Health, Noel Nutels. So Fábio, thank you so much for being here with us. And we will be discussing your protocol about Sanger sequencing of SARS-CoV-2 spike protein and the associated lab protocol that you publish in PLOS ONE. So with that, I would like to start by you briefly telling us what the protocol is about, how to use it, and why it is relevant.
Well, thank you for the invitation. And first, I just would like to apologize for my English. I might have be a little bit of problem forming some phrases, so please be patient.
No, absolutely. But like, I'm not a native speaker either, so good luck to the listeners.
So this protocol was designed to sequence a specific gene from SARS-CoV-2 that encodes the spike protein, the virus receptor. Why? Of course, using Sanger technique. But why is that important? Well, during the pandemics, it was very important for all countries to gather genomic information about this virus, to track the possible mutations, and to understand the virus evolution. And this was mainly done using new generation sequencing. With these techniques, these modern techniques, we can sequence the whole genome of several samples at once, and we can gather a lot of information, a lot of genomic information about this virus. However, what's the downside of new generation sequencing? It's very expensive, especially in developing countries. For example, if you go to a genomic database, for example, GISAID, where most sequences are deposited, you can see that the vast majority of sequences from SARS-CoV-2 comes from developed countries. And so with that thought process in mind, we aimed to design a protocol that it was feasible for any laboratory with a background in molecular biology, because Sanger technique is very widespread. It's easy to any laboratory to use, right? And even when you don't have the equipment necessary for Sanger technique, you can hire services from a nearby institution, or even how can I say, companies from international companies. Sometimes I use it as well. So, and why the spike protein? Why the virus receptor? Because this is the protein which this is the gene where most of the most important mutations tends to accumulate. Because this is very important for the virus life cycle, and is also the target for immune response. So it suffers a very high selective pressure. So this is, if you can't sequence in the whole genome, at least you can focus in the most important gene. Okay.
Yeah, thank you so much. Yeah, that was very illuminating. So, I mean, I understand that the motivation for developing the protocol was mostly to make a protocol that would be accessible for low resource settings.
Yes.
Was there any specific tweak that you have to make to the very well-known, very well-established Sanger sequencing for it to work in your particular sample? In this case, the spike protein of SARS-CoV-2.
The most important part is the oligonucleotides, to be sure they anneal properly to the how can I say, to the sequences that you're using. You have to test it from different variants that are circulating to see if a different variant have a mutation on the site that you use, that you design your oligonucleotides for. So yeah, this is the most difficult part, I guess. But it's something that any laboratory could have done as well.
And so, but once you had your protocol, what was the motivation to have it peer-reviewed and published as a formal paper in PLOS ONE as a lab protocol?
Well, the peer review process would allow us to detect any possible flaws in our protocol. So we were curious about that as well. And publishing as a peer-reviewed protocol would also allow us to share the protocol for other groups that could use it as well.
And elaborating on the decision to peer review, how did the feedback that you received from your peer reviewers shape your protocol?
Oh, we were very happy that they didn't find any major flaws in our protocol. We were very happy about that. And mainly they were worried to ensure that the protocol was easy to understand and accessible, right, to everyone. They used to correct some things like such as wrong pronunciations and such things, right?
So did you receive constructive criticism that helped you present the protocol in a clearer way?
Yes, to make it much clear, as clear as possible, yes.
And so, but why did you decide to make the peer review history public? Because it is contained in your article as well.
Oh, yes, of course. Since it could be important for us to determine if there was a possible follow-up in our protocol, we also thought that this information about the peer review history could also help other people that were using our protocol to improve their own protocols. So it was a matter of transparency. Yes.
Great. And now going into the details of the protocol itself, is there anything, while you were optimizing the protocol to make it work on your specific gene, is there anything that you tried but didn't work and therefore didn't make it to the final version of the protocol?
Well, yes. During the process of writing the protocol, it was in the middle of the pandemic, and it was very fast. New variants were emerging all the time and disappearing all the time. So we didn't have time to test all variants. So we have to focus on the variants that we have in hand, right? If we try to rewrite everything, adding new variants and testing everything again, we would never finish, right? Because it was very fast.
And is that because the oligos have to be very specific? Is that the reason?
Yes. We have to check if they are still annealing to new sequences.
And since the time of publication, have you had to troubleshoot? And if so, how and why?
Yes, at first, we designed 6 sets of primers of oligonucleotides, but we were having troubles to sequence the edges of the gene. they were not very good. So we have to design other 2 sets of primers to cover these edges. So we have the whole gene completed.
And if you were giving advice to the readers and those who would be using your protocol, what do you think are the most critical steps in the protocol?
Conservation of your sample, because some samples are irreplaceable, right? You can't have more of them. And conservation of the RNA after extraction, because RNA is a very sensitive molecule. So if you leave it, you might degrade, right? You have to be careful with the concentrations of your reagents, right? Because it will affect the effectiveness of your PCR. And you will also need to be sure not to contaminate clean areas with amplified materials, because it's very hard to decontaminate, and you might not be able to use that facility to perform that PCR ever again for that specific PCR.
I see. But
that's a type of knowledge that any laboratory should know with this type of background, right?
Okay, and but if I understood correctly, the protocol is very specific, or at least the oligos are very specific to the variant that you want to identify. Is that correct?
Actually, no, it's not specific to the variant. It's just to get the spike protein. We wanted to see if they cover every variant, right? That's the point. But actually, we try to check if the primers were getting the spike protein from Omicron variant. And one of those primers, of those oligos, didn't anneal very well because there were deletion exactly in the site of one of those primers. So we couldn't use that specific primer for Omicron, for example, right?
So
but the others were working.
Because I wanted also to ask if you have optimized the protocol further since publication, and it seems that you are either that you already did it or you are doing it for Omicron. Is that correct?
We checked for Omicron because it had a lot of mutations, right? So we find this, but we didn't think it was necessary to change, because it was only 1 or 2 primers that were not annealing very well. So we could rearrange the primers to get that part. And of course, if other people can also use those primers as a starting point, and they can design their own primers based on that as well, depending on their goals. So we didn't think it was necessary to change specifically, because those primers were still viable for previous variants as well.
Thank you, Fabio. And now moving back again to a more general question, I would like if you can share with us what has been the significance of publishing the protocol and publishing the paper for your career and the career of your colleagues.
Oh, it was very interesting because it increased the range of attachment to other groups, right? I don't know if this is the correct phrase, but for example, the applied mathematics that was also a collaborator in this paper, we are now performing new projects with them as well, right? We are trying to understand how the circulation of SARS-CoV-2 happened in the Rio de Janeiro counties, based on the number of deaths and the circulation of people as well. So it allowed this approximation. I'm not sure if that's the correct.
Yes. So it was like the starting point to start like further and future collaborations.
Yes,
with
exactly.
the groups that you prepare the paper with.
Yes.
Okay, thank you so much. And before we finish, I would like to know if there's anything else that you would like to add to this interview that we didn't touch.
I'd like to thank you again for the invitation, and it was very interesting to find about this platform, this protocols platform, because we now have a new tool not only to find new protocols, but to find everyday protocols, to know what people are doing about everyday protocols to improve them as well.
Oh.
So we can have some other hints about that too. So it was very interesting to find out about this platform.
Oh, thank you very much. And I'm happy that you find it useful. So with that, I think that we will finish. Thank you so much again, Fabio. I hope that you have a good rest of your day. And
thank you.
Thank you.
Thank you.